Cool. Okay, well now I'm excited. We're going to be dancing. Okay, so now we're moving on. And what we're going to do today, you're going to like this section better than the first section. First section all over the place gave us the basis. This is going to be sort of flowing. And we can already flow through the first half of the chapter. Hopefully, if you did some studying and listened to me on the bus, we should be able to do this. And we've kind of made it all the way to the end. So we're going to quickly review that and the exponential. And then we're going to continue on, basically, past the synapse. The people that had lab last Wednesday know that in lab, we actually what we're going to do a lot in lab is actually applying to this. You're going to need to know for the test. And it has to do with drugs and their effect on neurotransmitters. And so it's going to literally be flowing from this today as well. And some people wanted to watch the drug video again and save for that part, and that's fine. If you got time, we have room there to actually watch that. So right here, the circle of life, you're going to see this helps out a lot. This is sort of the ion distribution of these basically 4 ions that's going to help us throughout. What is inside the neuron? What's inside the cell? Negatively charged proteins and high levels of potassium, because we're going to show you how we made that. Overall, the inside is what compared to the out? And about for us, we're going to see later other neurons and muscles might change that number a bit, but it's basically the inside is negative compared to the out. Negative proteins in here, potassium's in here, let me move these over, and overall the circle of life has 3 ions out, which are what? And chlorine or chloride. Okay, so now when it comes down to it, those 4 ions we'll see today throughout this unit and the next unit, cardiac, etc., are going to be just opening and closing these channels. And electricity for us are these ions, one of these 4, depending on which ones we're opening, moving across the membranes. Okay, for all cells, muscle, nerve cells, everything capable of conducting electricity. That's why we get some basic chemistry, are those ions. Okay, so that sort of sets us up. Now, I wanted to talk about, and I wanted you to remember, as far as when we say negative 70, before we've activated anything, we call this the resting membrane potential of a neuron. I'm just going to put resting, because we're reviewing this quickly. And I said there were 2 main things that I want you to know of what set this resting membrane potential of minus 70. 1.
The potassium.
Yeah, sodium potassium pump is a major worker here, and it's pumping 3 sodium out and 2 potassium in, using a lot of energy. And so already, if you look at that again, by putting 3 positive out and only putting 2 positive in, the inside is becoming negative. But even a more important thing that actually creates this has to do that we've created this. Okay, we've put the water in the dams, but we've got a leak. Okay, there are leak channels which allow things slowly to leak down the concentration gradients. And of course, right now we're not dealing with anything but sodium, potassium. If we have a leak channel, sodium will leak which way? Down its concentration gradient. And that would make the inside of the cell more positive. Potassium is going to leak out. As potassium leaks out, it's going to make the cell more negative. And so you can go into the Nernst equation and all this math, but overall I just care that we understand the bottom line is. And the bottom line is that basically for the resting membrane potential, the leak channel, the cell is more permeable, to use sort of the correct term, I guess, to potassium, and then I'll put the words that make sense, compared to sodium, which means more potassium leaks out compared to sodium leaking in. Because leak it is a leak channel. Instead, if you don't like sort of the word permeable. So we create the gradients, sodium potassium pump. We have basically those 2 water in the dams there ready to go. But we do have a leak, sodium leaking in, potassium leaking out. when it comes to equilibrium, and I kind of showed the higher math of it and stuff, basically the bottom line is that more potassium's leaking out. It's a bigger hole, more potassium's leaking out, and that's creating this minus 70. And so, those are the 2 points of resting membrane potential.
Sorry, when you say 3 out versus 2 in, are you talking specifically about the potassium?
Yeah, for this one, while we're doing the sodium potassium pump, it's literally pumping 3 sodium out while it's pumping 2 potassium in, and it keeps doing it. So it's not pumping equal amount of charge out and in. Every time the sodium potassium pump works, 3 sodium go out, 2 potassium go in. 3 sodium out, 2 potassium in. So that means already we're making it more negative by putting more positive out than we are putting back. We go to the neuron. So we're going to get to the point of firing an action potential, which is what we did. First of all, we sort of had 3 parts of a neuron. What are my little hand-like, star-like guys? Dendrites. And what are they doing?
They're receiving signals.
They're receiving signals, and we're going to talk about even on the other side, they're going to receive signals. They're very branched because that increases
Surface area.
Okay, and let's say you really now, you're hitting on physiology, okay, so you're really studying this. Do these have some plasticity? Can we actually get more input?
Yes.
What do we have?
Spikes.
I know, they are like spiky. They're called dendritic spines. So then literally we can have new connections that actually, innervate the spines themselves. And so, and again, I read an article recently where during wake and sleep, they actually find that the dendritic spines basically will change even at that level. You're more awake or you're more active in one place than the other. So it actually, it's actually that plastic. And if you look at long-term memory, you actually see that, if you keep using a circuit more and more, you have more spines, so you get more information in. So there's some plasticity to these nerves, which is cool. Okay, cell body's got the nucleus and all that, and of course this is the axon. And one important part of the axon is the very first segment, which is the trigger zone, or the axon hillock. And so that's going to be basically the all or nothing place when we do the next part, which is graded and action potentials. Again, all this is review. This is how I recommend studying it on your whiteboard, kind of just going through this several times. So, again, when we're talking potentials, we're talking opening channels, ions flowing across, that's changing the membrane potential, the difference between inside and out. That's what we're talking about. Now, again, when we looked at this, 1, 2, 3, 1, 2, 3. We said there were 3 main differences between what a graded potential is and an action potential. And one of them, I just put third, is where we drew them. So graded potentials only occur in the...
Before the trigger zone.
Which are the dendrites and cell bus. And again, the axon is at, or the action potential only occur in the axon. And again, it is going to be the trigger zone, that's going to be the key. All right. So the other 2 features. So graded potentials. What were the other 2 features?
Variable strength.
Variable strength and
Losing strength.
it loses strength. Exactly. Loses as it travels. It loses strength. Again, you've had this before, so I'm not going to rewrite this. So, but let's see what this means again. What this is saying is, if we have, let's say, a weak signal versus a strong signal, whether it's a whisper and a shout, or a dim light, a bright light, anything we're going to be talking about in chapter 10 coming in. The key is that signal will open up some channels and create some sort of change in potential that we're sort of just drawing like that. If you have a greater signal, it's going to open up more channels, and you're going to have a bigger potential. So the graded potentials are related to the stimulus strength. The stronger the stimulus, the greater the graded potential. It's going to be sort of directly proportional, because the stronger the signal, the more ion channels they open, and boom. Okay? As it travels, they get smaller and smaller. And we talked about two reasons why, which were the ever-famous cytoplasmic resistance. Okay, somehow the cytoplasm is, because its resistance is sort of taking the punch out of it. Again, I'm not going beyond that because that's physics I don't even really understand. I haven't done that in a while. And the other one is leakage. So those are the two reasons that you sort of lose strength. But the real key for me is the understanding that the greater potential is proportional to the signal strength. Okay, so remember that. That's important. Boom. We have action potentials that these two bullet points are the exact opposite. Okay, so they're not a variable strength. They are same strength.
And size.
So it's going to be basically all of them are going to have same shape and size, which of course is going to be that guy over there. And the second bullet point here would be
Does not lose strength.
does not lose strength. So we're starting out at minus 70. If at the trigger zone we reach threshold voltage, which is in this case minus 55, if we reach that, then we're going to have an exponential. And the fact is that, and we can kind of, in fact, let me show that one. I kind of like this. So the idea that when we say it doesn't lose strength, it's not like it has this huge one wave that comes through. The bottom line is that it's like you have channels all along here, so that as one channel opens, it triggers the next one, and it's like dominoes falling. So because we have channels all the way along here, basically, and we'll go how an exponential works, as this segment gets triggered, it triggers the next, triggers the next, triggers the next. And that's sort of why that this one's different here, okay, for both of these. That's why it's the same size and shape, because we have the right amount of channels here, and we have them all the way throughout. We'll talk about variation, so it's not going to lose strength. They're also not very leaky. Yeah.
This might sound like a dumb question, but that's actually just a solution that passes through that has an electric, like, what is that, like a solution that?
These are, well, we're going to do this. This is obviously extracellular, intracellular, right?
Yeah.
When we open channels, we're going to have dissolved in water, in the fluid, sodium ions come in, and we'll see this potassium ions goes out. So it is literally a solution, the flow of ions across the membrane. Okay? That's why we were big on salts, and we made up the extracellular fluid. I mean, we had all those salts, because I knew what was coming is everything we talk about, nerves, muscles, etc., literally would be opening these channels, mainly these ions, these electrolytes, opening them, and then they're going to flow across. And that's what bioelectricity is, the flow of these ions across the membrane, in the fluid that it's in. Okay? So, that's kind of where we were. And you can see that starts to flow pretty nicely. So the next question I have, because I don't know if I asked this question, is, it's pretty fast. How can we speed it up? When we can actually, if we're comparing nerves, and we want to compare a nerve that fires faster, that reaches the end faster, so it has basically see how do I want to say this, a faster conduction rate. So for timing, let's say it reaches threshold, and we're timing from beginning to end, and then we time another one from beginning to end, some will go faster. They have a faster rate of conduction rate. And there are two features which relate to that. Do we know what they are? One is myelination, which I'm going to talk about more, and the other one is a larger diameter neuron, or axon. So, and really, the larger diameter is saying if you have, instead of, and we'll talk about diameters, instead of having a given diameter, if it's larger, that's less resistance. If you just even look at, water going through a pipe, okay, if you if you enlarge the size of the pipe, this is less resistance, right? And then it can start flowing, faster. This will be how blood works. Physiology is based upon chemistry and physics when it comes down to it. Because that's what we're based on. We obey those laws. That's why if you come with a chemistry and physics background, it makes a lot of sense, to you. If not, it's still going to make sense. So, yes.
So when you talk about larger diameter, you mean in the dendrites?
No, I mean in the axon.
Oh, in the axon.
Okay? So the idea is, because that's where the action potential is really happening, right? That's only that. So if you have a, 3 millimeter, it's going at a certain speed. If you have another axon that's like 7 millimeters, okay, that's less resistance, that's going to travel faster.
Okay, so axons are different sizes.
They're different sizes.
Okay.
They're different diameters. And so if you have a larger diameter, that will go faster.
Okay.
Now myelination, here's the idea of myelination, is instead of having channels all the way through, like we did here, what we're going to do is myelinate. And again, we don't basically wrap that myelin around the whole thing, so it's like a wire, we don't completely wrap it. We leave spaces open that we actually then pack those channels all together. So we're not going to have channels going throughout the entire axon membrane. They're going to be packed in the spaces that don't have myelin, and we call those spaces
Node of Ranvier.
the nodes of Ranvier. Okay? So I like to say that. So the nodes of Ranvier are the unmyelinated regions, and the idea of this is, okay, because it's actually up here. So instead of having to open up all these membranes like the dominoes, we've got them packed in this thing. So once we activate, in one particular node, we're going to have a rush of that ion. Okay, I forgot I have to actually do the action potential. That will then trigger the next segment. That will trigger the next segment. There's no leakage at all, kind of in this myelination region, because that's like membrane. So you have a rush of sodium in, and depolarize the next set and the next set, and it increases the rate by up to 50-fold. I sort of like to demonstrate this, just because you know me, I like to jump around. Let's say I have to get from here to the end of the desk without myelination. Okay, well I've got 10 doors to open. Okay, so 1, 2, 3, 4, 5, 6, 7, 8. Okay, but if I'm myelinated, I have 2 doors. 1, 2, faster. So that's sort of the idea. Instead of having the domino one where we have to open up channels constantly, by packing this region, and then having the region in between myelinated, we are able to basically have this sort of jumping thing called saltatory conduction, which is quite a bit faster, okay, up to 50-fold faster than a neuron, of the same size. You know what? I just realized this one is in the peripheral nervous system. So who myelinated him? What cell type? Schwann cells. I made a mistake. This one's in the CNS. Who myelinates CNS neurons? Oligodendrocytes. Oh, well, we said astrocytes. Astrocytes are associated with blood-brain barrier. We need immune cells. Who does that? Microglia. So, again, what I'm trying to do is, when you study these things, don't wait 2 weeks from now and start this. Every day, start to kind of do this and just remind you, and then they're formed, okay? So that's kind of what we did before. One last thing on myelin. So when you look at demyelinating diseases like MS, you have this idea that, it's going to be one of those sort of autoimmune type things where you start to destroying your myelin sheath. Now, these axons don't have channels all along. Okay, they used to have this wrapped in. So the idea is now you're going to have this idea of a lot of the sodium, et cetera, leaking out, and maybe you're not going to get as many, you're not going to actually complete the action potentials, okay, as often. And it's progressive. So the more myelination you lose, the slower it is. You start losing conduction. If you're trying to control muscles, you're going to have less control of your muscles. So that's, demyelinating diseases, how they work. You're losing this myelin. And this myelin is important for this quick conduction. Action potential. Forgot to review that before we added that little thing. So if you look at the action potential, we're gonna come back to this refractory nightmare. I promise you. So if you look at the exponential, it has 9 things based upon Silverthorne, but we don't want 9 things. I want us to remember 1, 2, 3, 4, 5 things. 1, 2, 3, 4, and 5. So we reach threshold and we have that peak going up. What do we call this process?
Depolarization.
Okay, in this case, okay, then we go back to the circle of life or in your brain. What is causing that depolarization? And sodium goes in. I always like you to go flow. That's why you can always use this so you'll remember. If you open up sodium channels, it goes in. That makes it more positive. That's what's happening here. So, again, depolarization, sodium channels open, sodium goes into the cell. Okay? 2.
Repolarization.
Well, at 2 right now, we're just at the top. So we sort of said sodium channels closed. If they kept open, maybe we'd even get more positive, maybe. But we're gonna make this complicated in a second. But right now, we basically said sodium channels closed, so that no more positives are going to come in. Now, at the same time that we triggered the sodium channels, we actually triggered another ion channel, but it was slower. And so what are we calling number 3 overall?
Repolarization.
And that's being caused by what?
The calcium, potassium channels opening.
Yeah, and potassium is going
Out.
out. So in the exon, we're only using those 2 ions, sodium and potassium. Don't worry, we'll get these others involved. Repolarization, potassium channels open, and you can see on there, it makes sense, potassium will go out, making the cell more negative, and that's repolarization. Up to we get to minus 70 again. Now we dip below this, and what do we call this below?
Hyperpolarization.
And what's causing that?
More potassium's leaking out.
Yeah, we're not going to call it leaking out. I mean, it's just a minor point. It's flowing out because these are the gated channels versus leaking channels. We're going to get to leaking here, though. So hyperpolarization, again, these channels just stay open longer. So potassium, right now I'm just going to make it, keeps going out. Okay, now we're hyperpolarized. Again, I just don't write. Obviously, the potassium channel is closed. But number 5 is what's going to cause us to slowly get back up to resting membrane potential.
Sodium leaking.
Now that's leaking. And so number 5, which isn't in those 9 steps, but it's below, sodium will leak back in. That's why it's slower until we get back up. It's still milliseconds. It's not that slow. But until we get up to resting. It might even make sense, okay? Because if you look at this, minus 70 attracts sodium to a given degree. If we're at minus 90, okay, negative 90, that might suck the sodium back in on through that leak channel, right? Because positive and negative attract. So maybe that we're hyperpolarized helps us to get sodium in until we reach that resting equilibrium. Okay, then everything's just like homeostasis. We have kind of this equilibrium. And that's what I want us to know about the action potential right now. Okay, those 5 steps. Again, all we've been involved are sodium and potassium channels. Sodium channels opening, going in, that's depolarization. They close, yeah, we got to talk about that. Then we have this idea of repolarization and hyper, potassium channels. Potassium goes out. Then we have sodium leaking back in, we're back up to resting. And that's the action potential.
On that side, that happens in the axon.
Oh, this is only in the axon.
Okay.
Okay? Now these other things are, we're going to talk all the different channels are kind of opening and closing up here, and that we'll talk about that in a way. But overall, yeah, this is in the axon. This is what action potentials look like. They're only in the axon doing this. And it's basically in this case, only these 2 ions involved.
So for the rated potential, it doesn't matter how low it gets, as long as it gets to the trigger, it's going to, all this is going to happen.
As long as when it gets to the trigger, it's minus 55 or higher. So overall, if we start at minus 70, it needs a depolarizing stimulus to reach the trigger zone that's sort of plus 15, right? If we're minus 70 plus 15, we'd be at minus 55. And these are voltage-gated channels. You reach minus 55, boom, this all happens. So this happens. If you don't, if you only get to minus 56, nothing. It's not like a little one, okay? It's an all or nothing. If we had a ton of stuff and we actually got to like minus 30, it's not bigger. It's going to be just that size. So unlike this, and we'll get to this later today or next time, unlike the graded potentials, which are proportional to the signal, this is all or nothing. You know, it's either on or off. Okay? And we'll get to how we figure out what happens down here.
So it has to be a negative 55 to shoot, not negative 70?
Yes, negative 70, it's like, that's its
That's
rest.
equilibrium.
That's equilibrium, that's what we call like the resting memory potential, resting.
Okay.
So they've triggered these, and we're going to look at these voltage gated channels. It has to be minus 55 or more positive. That's the threshold.
And the nodes of when those are only present when Wyland's
Yes. And those ones are where you have, again, instead of having them sort of all the way along, you have them, like we're kind of showing here, you actually have, them, not demyelinated, just all packed within the nodes, okay? And the myelin doesn't have it. An unmyelinated nerve, not a demyelinated, but one that's not supposed to have it, has these channels spaced all the way along. Take a drink. Okay, we have one complication. This is a flow, and then we're going to get back to flowing. So if you looked at this, if you sort of looked at what we did and how you could study, you could see you could literally start with a circle of life. What created this? What are graded potentials? What are action potentials? Okay, what defines the action potential? Okay, how to make it faster? Talk about myelin. Okay, and then in a minute we're going to talk about the end. The release of neurotransmitter and the effect on the postsynaptic cell, which is the end of the chapter. Okay, so literally this chapter will flow sort of in one, big whiteboard or one type of thing. And if you study it that way, this is not a note card thing. I'm going to study this and study this. If you study it this way, you learn it and you're going to remember it for the test and for anything else. So I really recommend. This is a good chapter for this. It's nice, flowing. Except for this one little thing which we've got to talk about, which happens to be this. And I'm going to go back to what this is. And we'll keep that up. So when we look at number 2, and we'll see the picture on this, and we want to look at that sodium channel, okay, that's opening and closing. What we're going to see is, we'll come back to those words. What we're going to see is, first of all, when you see this plus minus, that means this is a voltage gated channel. There was chemical and there was mechanical. This is voltage gated. Once this reaches minus 55 or more positive, this will open. Right now it's closed. But look at this channel protein. It's got this activation gate that will be activated by, voltage change, but it's got this guy here called an inactivation gate. So I'm doing two things here. I'm going to show you how it works, and even more importantly, I'm going to tell you why, because that's what I really care about is that you understand why. So we're looking at this, and we're at minus 70. We're following it over here, and we're going to reach threshold. Okay, when we reach threshold, just like we said, in this case, it's the activation gate opens, sodium rushes in, depolarization, just like we said. Now, here is going to be the little bit of difference. When we went to number 2, and we said, I erased it up there, but we said sodium channel is closed, it's not that one gate, it's something called the inactivation gate that closes. Okay? And as long as this is closed, we don't care what the voltage is. This voltage might be fine to let sodium in. We're not going to let it in. And that's called the absolute refractory period. So no action potentials are possible because you can't get sodium in there, no matter what the voltage is. Again, it's sort of said that was the reason why. Now, this basically corresponds to, and for me versus the book, basically.
Is that only on the voltage channels, or do all the channels have it?
Just on the sodium voltage one.
Oh, okay.
So we're only talking just this guy right here. Okay. there might be others somewhere else that has this, but really we're just talking about this that has the two gates. And so basically that gate is closed for the most part basically during repolarization. So that represents, again, the picture they'll have is a little confusing, and I don't like these pictures too much, I don't really use them. But the idea is basically they go a little further. But basically, this is when the inactivation gate is shut. Okay? This represents the absolute refractory period. Absolute. Okay? When, again, no X potential is possible because no ions can go through as long as this inactivation gate is closed. Then it's going to open. But first I want to tell you why. Okay? The reason why. That's sort of more important than even the mechanism to it. So, let's look at the flow. And then we're going to see. So we had this gradient potential come in, it reached threshold. Remember, this is minus 70 or so, but we've got sodium coming in, triggering these voltage gated, and boom, sodium's coming in. Okay? Boom, more sodium's coming in. Now check it out here. Well, we can even check it out here. When sodium comes in, does it know we want to go that way? Does it have like a little brain or something? How many people say yes? No. It can go both ways. Now the question to us is, well, if sodium's going this way, it could trigger those things. Do we want action potentials to travel both directions?
No.
So, here is why we have the absolute refractory period. So, as we trigger the zone, sort of the next one, although sodium can come in both ways and make it so that these channels would be open, because the voltage is good, we're not going to let the sodium come in. We're not going to let action potentials travel backwards during that small time right behind it. That's the refractory period. So in this case, the refractory period, we have this idea where no action potentials are possible because we have the inactivation gate closed. Even if the sodium makes it so the voltage guy is open, we're blocking it. So again, that's to prevent action potentials from traveling both directions. Then as you get to the next segment, this thing is reset again, and again, there's no more sodium, there's not enough sodium to trigger it, so we can have another action potential. So we go through this thing right behind this as we go. This refractory period, again, is when that inactivation gate is shut so that the action potentials only will flow this direction, even though sodium can kind of flow both.
So it's to prevent the sodium from flowing into the cell body?
It's going to flow in, but what we don't want to do is if this sodium comes in and there's a sodium channel there with the activation gate like there is, we don't want that to basically allow more sodium to come in over here. Because then what it's going to do if it keeps going, well what? It will trigger more sodium. It will literally trigger the action potential from going backwards.
Okay.
So we have that little time right behind it in which we've got that. We've got this thing shut. Okay? So during this whole repolarization, even though sodium's coming in, let's say over here, so that now this would be open, boom, we're not going to let that happen. And then it resets. By the time it resets, we're way down there, right? This is pretty fast. So now we can get another one going this way. So again, it doesn't flow as nicely as the other things, but we sort of have to understand the overall. That the absolute refractory period is when no X potential is possible. Okay? I'm basically giving it toward repolarization. That's when this thing is closed. And again, the bottom line is to prevent action potentials from traveling backwards. Before we go into the fun, relative refractory period. The relative refractory period, and you can just even look up here, I almost want different chalk, is, okay, at this point, when it's hyperpolarized, okay, the gate's reset. Okay? But we call it the relative refractory period because if you're hyperpolarized, right, we're hyperpolarized, it's not going to take plus 15 to get the threshold. It's going to get what? Plus 20, plus 25, right? It's going to take more to reach it. So you can have an action potential, but it takes a stronger than normal stimulus. Here, when it was a minus 70, it just took plus 15. During this period of hyperpolarization, which are more negative, it's going to take more. So the gates are all fine again, okay? So that's not part of this. But the idea is, because we're hyperpolarized, Okay, and again, don't worry about this excitability thing, because we're just not dealing with this level of it. But the idea is that when it's hyperpolarized, it's going to take a stronger than normal stimulus to get the threshold, because instead of just needing, again, this, it's going to need, this, or this. It's going to need much more.
What would the stimulus be?
The stimulus is just like everything else. The stimulus is all these inputs coming in, and are we going to reach how much that's going to add up to. So now we might need this trigger zone to add up to plus 20 or plus 25 in order for it to fire. Just for, this is milliseconds. So we're talking a couple of milliseconds. It helps us time, our action potentials, at a certain rate too, by having it where, it's not, you have some sort of space here before you can get back to sort of normal. But during this period here, no action potential is capable through that segment because of that inactivation gate. Then it just takes a stronger than normal stimulus. And that's the idea of these two terms. Okay? But we're ready for something more fun. So that's like a diversion I sort of hate, but I have to do it. Because now we're going to clean this up. And you know who's been unhappy? Very unhappy, actually. Calcium. Because we haven't talked about calcium. And calcium is so important. It knows it's important. It's got a little kind of, ego. It's like, your heart won't beat without me. Why aren't we talking about me? We're going to talk about calcium now. Okay? So let's erase some of the mess. Let's clean this up. Okay. So we made it to the end, right? We just did the action potential, but we've got this synapse here. And what are we going to release? What's sort of the idea of trying to get across here? We have to release neurotransmitters, a chemical that's stored in the nerve terminal here. Okay, we'll talk about the different ones, serotonin, dopamine, etc. So they're there. We need to get them released. Guess who's going to be involved? Calcium. Come on, where are you, calcium? Okay, so these two ones here. So overall, again, we have this idea. We've actually made it all the way to the end. And now, sort of in the nerve terminal, we have that neurotransmitter that has to be released, bind to the postsynaptic cell, and do its thing. And so it's a chemical synapse. We have signaling electrochemical, depending on how far we're going. So, calcium. So here's where calcium comes into play. That at the axon, we're just dealing with sodium and potassium channels. In the nerve terminal, we have voltage-gated calcium channels. So this actually, by the way, these are pretty good as far as like going through a process here. I'm kind of just saying what's up here. So we're going to have what we've just described. Action potentials travel down the axon, but when it reaches the terminal now, we have voltage-gated calcium channels activated. Just like we had sodium and potassium up here. And again, you look at the circle of life. If we open up calcium channels, calcium will go into the cell. And what it does, by sort of a complicated process we're not going to talk about, it causes these vesicles to fuse and release or exocytose the neurotransmitter into the synapse. That's how we get neurotransmitter out of the nerve terminal into the synapse. It's finally using now calcium, voltage-gated calcium. It's triggered by the action potential, okay, because that changed the voltage, but now these are the channels. Calcium comes in and basically causes this to fuse and release the neurotransmitter into the synapse. And then it's going to bind to a protein on the postsynaptic cell that we're going to talk about and cause some sort of cellular response. Maybe another nerve firing, maybe a muscle contracting, everything that we're going to eventually talk about. It's going to cause something. And so these 5 steps, we make it finally from the beginning to at least this next step here, the release of neurotransmitter. That's the important one there, calcium. Calcium's getting happy. Calcium causes the release of the neurotransmitter here at the nerve terminal. It's pretty good. We've got a flow here. Now, again, if you've had the drug video already, this next part sort of is kind of a review. We're releasing neurotransmitter, and we'll use this slide here. You know, neurotransmitter is hitting these receptors. Do we want the neurotransmitter to stay there forever?
No.
And keep hitting them. We want control. And there's 3 ways to control it. This third way is sort of passive, so I just want to say it first. That's diffusion. So the neurotransmitter that's in the synapse can diffuse onto the blood and have less effect. But these two are big. They're going to be involved in normal control and in drug control. And the other 2 ways we control that then are basically reuptake pumps and 2, enzymatic degradation. Those are the 2 main ways that we maintain some sort of homeostasis between sort of, the stimulation and the effect. Okay? We want to control the amount of neurotransmitter that's there and for how long. So, we have this idea, number 1 is this idea of reuptake. We have reuptake pumps that will basically, once the neurotransmitter is released, it's kind of doing it, but it's also reuptaking it into either glia or the cell body. We can reuse it as well, it gets reused. But the bottom line is that helps us control the levels of neurotransmitter by reuptaking. So we're releasing it, but we're reuptaking at a certain rate to keep the right balance, hopefully. The other way is shown in number 2. The other way is that we're going to take sort of, the amount that's released and basically use an enzyme to chop it up, okay, at a certain rate. So we're controlling, again, how much is stimulating and the effect by controlling the levels of neurotransmitter for how long. And those are the 2 that we're really going to talk about as far as what goes wrong, let's say maybe some conditions, depression, bipolar, and sort of what drugs are used there, as well as some drugs which aren't prescribed, like cocaine and heroin. We're going to do all kinds of drugs for people tonight. We're not going to do them, but we're going to talk about it. Like the whole board, we're going to talk about the action of like 7, 8 drugs. It's like drug night. So that's why, come on back for another one. Even if you were on Wednesday and you want drug night part 2, see it again. Okay, you're going to learn almost every drug you might want to know. We'll learn tonight. But most of them work basically at this level, sort of manipulating this. So those are really 2 important. So there's diffusion, but there's really these reuptake pumps and enzymatic degradation. And the video really shows this well.
Is number 2 the enzymatic degradation?
Yeah, enzyme inactivates neurotransmitters, so it's chewing it up. Pretty good. Okay, we made it to neurotransmitter. What the next 2 slides are, they're not on your handout, but they are on the PowerPoint. And actually I'm going to update the PowerPoint. These 2 slides, which are here and here, if you look at the bottom, those are the neurotransmitters that I want us to know. And so let's look at those. You don't need to know the structure, which is shown up there. And when we look at neurotransmitters, there's a ton of them. They end up being sort of very small molecules. A lot of them come from amino acids, okay, which we're kind of showing here. Now, let's look at these 3 things I have here, and so you have to know. So acetylcholine, which we will, the drug video will talk more about, but also what we're going to talk about is acetylcholine. A lot of times we're going to basically abbreviate it ACH because it's hard to spell acetylcholine. When we do basically, in chapter 12, the control of skeletal muscle, we're going to see that's going to be excitatory. That's what's going to be released to get your skeletal muscles going. When you look at the video tonight or again, we're going to see it likes to be controlled by enzymatic degradation using an enzyme called acetylcholinesterase, which you can see up there, right? A lot of times we'll just say, ACH, ASE. Enzymes will end in ASE, right? So it's going to be deactivated that way. So it uses that, and when you see the video, you see these little scissors come. It's very visual video. But the idea is acetylcholine, excitatory, and it's sort of controlled by this enzyme. You don't need to know this bullet point right now, but when we do cardiac next unit, you're going to see it actually slows down heart rate. So, it excites skeletal muscle but slows down heart rate because it's not the neurotransmitter itself, it depends on what receptor. And that's going to be sort of cool. But right now, that first line. Now, these guys. Okay, and really, we're going to talk a lot about dopamine tonight in lab, because that's a fun neurotransmitter. So all of these, dopamine, epinephrine, and norepinephrine, basically are called a class called catecholamines, and they come from the amino acid tyrosine, all of them. Just different enzymes will make slight differences to make these guys. You can see some differences between norepi and dopamine here. But they're all coming from tyrosine. And so all of them are called catecholamines. By the way, next unit with the cardiac, epinephrine is like adrenaline, and norepinephrine is like noradrenaline. That's the old term. So we're going to see a lot of that. But all of those are catecholamines derived from tyrosine. You need to know that. The third one that we're going to talk about in drug, is the videos as well tonight, is serotonin. And that comes from the amino acid tryptophan. And we're going to see that's involved in sort of normal moods and anxiety and depression and that type of thing. That's a very important neurotransmitter. So we need to know that page. Again, all this is on your PowerPoint, so you can see it there if you don't want to write all that down. But it's good to slow down. But I'm moving on. So because it's in your PowerPoint. So basically, don't worry about the nitrous oxide, but these 4 lines. So, and we're going to use this a lot. So GABA, you can see the structure up there, very small molecule here. GABA is the major inhibitory neurotransmitter. Okay, we're going to talk about that when signals hit the next neuron or muscle, it can either excite or inhibit. And we're going to view that next. It can make it, which means it can make it more likely to fire or less likely to fire. Because we're always sort of in a balance. We have positive and negatives coming in, hundreds of positive negatives coming in, trying to add things up. So the major inhibitory neurotransmitter is GABA. And I think somebody there already mentioned GABA when I was asking different neurotransmitters. So GABA is major inhibitory. The major excitatory, the major one is glutamate. Okay? So when we're looking at the CNS, there's a lot of them. But when we're thinking of just sort of the major ones, GABA will be the major inhibitory one we'll talk about. Glutamate is the major excitatory. And we'll definitely define that more in a minute. Okay? The next two will go together for chapter 10, okay, which we'll start on Wednesday, which is all the different senses. So one of the senses is pain. And I love pain. We're going to do a lot of pain stuff, okay? Pain is cool. So the major neurotransmitter involved in the transmission of pain signals to the brain, luckily, is called substance P. P for pain. Okay? So that's the major neurotransmitter that will be for transmission of pain. But we have some natural sort of morphine. We have some natural painkillers that we release, and those are called endorphins. And so they're going to modulate the perception of pain. The pain signal that reaches our brain and we feel a different level of pain, whether we take morphine or we sort of, bike ride 50 miles a day or run 5 miles a day, you can release natural endorphins, literally natural morphines that sort of, help you feel better, but also can diminish, pain perception as well. So substance P transmits, endorphins modulate perception. And you don't need to know this one for now. Again, we do cardiac noise. Well, guess what? We're here. Postsynaptic cell. Okay? We made it now to the neurotransmitter released, some examples of neurotransmitter. How about that effect on the postsynaptic cell? I'm going to come back to that. So, we got a crazy diagram here. We're not going to quite use, I'm going to put it up there a second, but we're not going to use it too much. Because you're going to be able to figure this out a lot easier. So, okay, I just started redrawing this again. We have sodium, calcium, chloride. We'll get chloride involved now in a minute. So there's 2 points I want to make. And then we'll definitely turn this crazy thing off. But one point I actually do want to show here on the side, and they're kind of there. When the neurotransmitter binds the receptor, okay, so we made it up to here, now we're kind of getting on the postsynaptic cell. When it binds the receptor, there are two basic classes. One's going to be faster than the other. The fast one, which again, when you see the video, you'll sort of see which one might be. When the neurotransmitter binds, it opens the an ion channel directly. Okay, so sodium will come in, electrical clone will refer, etc. So that's going to be quick. Basically, the neurotransmitter binds. It's a chemically gated ion channel. Boom, it will open fast. The slow one uses, well, we didn't cover chapter 6 as far as second messenger system, and that's fine. We don't really need it. All you have to know is that some of these work by when the neurotransmitter binds its receptor, it basically goes through an indirect way to open up ion channels. It goes through something called a G protein that changes these messengers and then opens an ion channel. So ultimately, we're opening up ion channels in the postsynaptic cell, but we want to distinguish sort of between fast and slow. Fast ones will open up the channel directly. The slower ones will use G proteins and go through this slower method. You're not memorizing all these steps at all. But you realize that when you use a G protein, it's going to go slower in order then to open the ion channels and see it.
What makes it like slow or fast? Like depending on what neurotransmitter is released, how do you know?
Well, you only know based upon when we tell you, or what they studied of how each one works. So you don't know offhand like which one would be what, but we're going to show in the video a couple examples. They determine different neurotransmitters just happen to use, some use the slower one and some use the faster one. So it just happens to be what they use. In the video tonight, we'll give an example of each. But right now, it's just the idea that will help on the video tonight, or remember the video from last time, that fast are going to be direct opening of ion channels. The slower ones will be ones that use this G protein and then open ion channels. Now, okay, well, I guess I'll keep it on a second, but not for long. Okay, but just put it there. Don't look at the picture at all right now. But the idea of this will be, and we'll kind of go on this. I want to clean up all this. This is like all of this. Let's get rid of this. And in fact, we got this neuron here. Okay, let's go ahead and do this. I'm just cleaning it up a bit. Here's our action potential. Okay, but now I'm going to talk about, instead of one presynaptic, neuron coming in, let's take a couple. And we'll see this right, because there's not one neuron going to the next neuron. There are hundreds of neurons going to hundreds of ones, okay? In fact, let's just look at a picture kind of like this. Okay, we'll come back. When you really look at the dendrites and cell body of a neuron, it's receiving hundreds and hundreds of inputs. Now, some of these, and we're going to draw it up here, some of these will be, when it releases, excitatory, which means it's going to depolarize, make it more likely. And I'm going to make like a bullet points here, right? So we're going to introduce it. Some are going to be excitatory, others are going to be inhibitory. Its neurotransmitter is going to make it less likely to fire. And so we call these overall, and we're going to be able to see them, either causing an excitatory postsynaptic potential. Okay, it's either going to excite, this is the postsynaptic cell, or it's going to inhibit it, make it less likely to fire. Okay, excitatory or inhibitory, EPSP and IPSP. So I'm going to put these here. EPSP, IPSP. Okay, excitatory versus inhibitory. So excitatory, if we just come back to this, is that going to be a depolarizing stimulus or a hyperpolarizing if it's going to be excitatory?
Depolarizing.
Let's write that. It's going to make closer to threshold. It's going to be a depolarizing type of stimulus, okay? Again, make the inside more positive. However you want to try to start remembering this. Versus inhibitory, If you put a neurotransmitter which is inhibitory to the postsynaptic cell, an IPSP, it's going to basically be
Repolarizing.
or even
Hyper-
hyperpolarizing. Okay, it's going to oppose it. So it's going to be something that's going to cause repolarization slash hyperpolarization. Make it less, make the side more negative. And so, kind of, you know, when I show this sort of symbol here, we'll actually see it. It's sort of, it's not a positive negative charge. It's just saying when this releases its neurotransmitter, it's going to be causing an EPSP. So we're sort of just making a little symbol there for that. If you have another neuron that I'm sort of symbolizing by negative, when it releases its neurotransmitter and it binds, it's going to be causing a, you know, a less likely to fire, of repolarization, hyperpolarization. So this is more likely to fire, less likely to fire. Now, instead of looking at this little, you know, I don't know, Rain Man thing, okay, I don't know what that is, we're going to be able to figure out from this what channels will open, 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 open. What channels will open that would be part of either an EPSP or an IPSP. And I'm going to show one little extra that we can actually close a couple of these channels that would also cause EPSP or IPSP. And again, I want to emphasize, every time I say again, I want to emphasize when I'm using the terms open and close, it's a simplification. You know, you're sort of, you know, increasing the permeability and decreasing the permeability, you know, making it more likely or less likely. It's not like it's absolute. You know, I just find it easier to talk this way, right? So you're basically sort of, you know, getting it more permeability or less. Now, we've already done this. We can do this right now, okay? So if we're talking something which is going to cause an excitatory postsynaptic potential, it's over here. What do we want to open?
Sodium.
You open up sodium. Sodium goes in, that's a depolarizing stimulus. You want to open up
Calcium.
calcium. Calcium comes in, that's a depolarizing. Let's forget the close right now. Let's go on this. If we want this to be repolarization or hyperpolarization, we're going to open up
Potassium.
and chloride. Exactly. We open up potassium. Potassium goes out, makes it more negative. We can have it trigger opening of chloride. Chloride will go in, make it more negative. Both of those are IPSPs, which make it means they would make it less likely to fire, less likely to reach threshold. These first two opening, as we did almost the first day last time, basically if sodium and calcium open, that's going to cause a depolarizing it, making it more likely to fire. Remember, these are causing graded potentials. Remember that we were going to add up at the trigger zone. So we're going to have some ones that release neurotransmitters which are excitatory and some which are inhibitory. What are they doing here? What channels are they opening? Boom. Now for closing, the only channels we can sort of tweak close or less likely are these guys. So we're not going to close these, you know, we were only opening or not opening, so to speak. But potassium and sodium, we've got a lot of control over. So if we want this to be depolarizing, which one do we want to sort of close? You want the inside to be what? Negative or positive?
Negative.
Okay? No, to depolarize.
Positive.
You want to try to get positive. So you want to close potassium. Potassium will stay in longer, right? So we'll close potassium. Also, you know, you're not going to open sodium and close sodium too. So once you get this makes some sense here. And if we want this to be inhibitory, we're going to close sodium, so positive doesn't go in. Okay, open potassium, close sodium. I mean, the closings you got to look at a little bit longer to get it, but it's always going to be the opposite. But again, EPS means more positive. We're going to let positive in and keep positive in. Okay, that's how they're going to work. For IPSPs, we want negative. So we're going to get the positive out, get the negative in, and stop positive from coming in, like closing that. And so the beauty of this is, instead of trying to look at this thing, okay, which is saying it's kind of the same thing, unless you like it, okay? If you're a Rain Man or a Beautiful Mind guy and you photographic memory and you're like, boom, I got it, I can use this flowchart. But what I recommend is that you don't have two separate note cards on this, that you literally practice going from the circle of life and be able to figure out which channels open and close to create EPSPs and IPSPs. Okay? That's how you're going to get it. You come to the test, that's not an essay question, that's stupid, but I'm going to have all the following could be EPSP except, you literally will sort of, you might have it in your mind, you literally write these things next to it, you find the right answer. Okay? So does everyone see how using this can get here versus just some random note cards, I hope?
So it's always what the inside of the cell is, whether it's negative or positive.
Right.
You're always looking at the inside.
Yes, you're always, because this is what's happening on the inside, those voltage gated. So you're looking at the inside. If it's becoming more positive, we might reach threshold and boom. If it's becoming more negative, you're less likely to reach threshold and you'll have no extra potential. We're literally looking at what's happening here at the trigger zone. These are opening and closing channels like we said way back when. Some are positive, some are negative, okay, excitatory or inhibitory, and we add this up here. When we add this up, if it's minus 55 or higher, we have it. If not, we've inhibited it. Remember, we don't want everything to be going like this, okay? I am a little hyper. We want to have plenty of inhibitory, okay? You cut a chicken's head off, what happens? They go around, I don't say you do that at home, but you've seen it, right? You lose a lot of inhibitory that's naturally there. So inhibitory is really important. We tweak this up and down. So look what we've done. We've gone from just how we actually create the resting membrane potential of anything, all the way to what happens in the dendrites, the cell body, the action potential, the release of neurotransmitters, the control of neurotransmitters, and the effect on the postsynaptic cell. Sweet. Nice and flowing. Any questions about that as far as what's the study? We're not done yet, but kind of where we are.
All those neurotransmitters that you put, those are all the ones we have to know.
Yeah, those are the ones you have to know. When we do the drug video, we're going to spend a little more time on acetylcholine and dopamine we're going to see. But yeah, those are the ones we have to know. Okay. A couple more little points. And then that's the chapter. Remember, chapter 8 is the base of this whole test. You know, I don't know, 40% of the test is knowing the details of this and being able to sort of apply it. Okay. I'm skipping around a little bit. I'm going to come back to that, what I skipped. I want to talk about potassium. There's 3 things left to do, so we'll go to you. Potassium levels in the blood, extracellular fluid, extremely important. We're going to be talking about that throughout this course. And anybody that's sort of already out there in the field sort of knows that you monitor potassium levels. Okay, that's very important for muscle function, nerve function, heart function, and literally we're going to build upon this each unit as far as its importance. Now, we have it here, and maybe since I'll keep that up for a second. And so, overall, we're talking nerves now. And remember, when we draw potassium more inside, well, there's some outside. Okay, it's not like there's none outside. We've just pumped so there's more inside than out, and now we have, at rest, sort of some equilibrium, where we have a lot more in versus out, because that pump will keep going. Okay, and so if you look at that, let's say we're normal kalemia. Okay, kalemia represents potassium levels. Then, if we have a sub-threshold stimulus, are we going to have an extra?
No.
If we have one which is above minus 55, are we going to have extra potential?
Yes.
Yes. Now, let's look at how this will change if we have either too much potassium, hyperkalemia, or if we have too little, hypokalemia. And we know hyper and hypo by now. So we're going to try to figure out how this is going to affect sort of the cell, and we'll kind of be looking at the resting membrane potential and the effect. Now look over here, we can even reason this. Okay, if you have, we have this balance, right? big K, little k, we had a certain balance. If we have hyperkalemia now, that means we put more potassium into the blood extracellular fluid than we had before. We have hyperkalemia, too high. Now, if this were in equilibrium, whether you lose long mass X or whatever you want to use, one or two things are going to happen once you do this. Either some of this potassium is going to go in more, or more of this potassium is going to go out. How many think more potassium is going to go in? Okay? In other words, if this were to balance, we were supposed to have this amount, and now we have this amount, that's going to go in and reach another equilibrium. Hyperkalemia means overall, some of this potassium that's too much out here is going to go in, and when it does, what's that going to do to the resting memory potential?
Make it positive.
Right. And so now, you're going to have, instead of the resting memory potential being minus 70 and you need plus 15, now it's going to be closer to threshold. So that sub-threshold stimulus, which wasn't supposed to do anything, boom, it's going to actually cause an action potential. You're going to have hyperexcitability. So hyperkalemia, bullet points. We have a higher than normal amount of potassium. You know, you ate 30 bananas. Or something else happened with the kidney and our drug effects we're going to see. Well, later we'll talk about ACE inhibitors for blood pressure. You monitor potassium levels because it's going to affect potassium levels. So potassium is big. Hyperkalemia, too much potassium. It's basically, again, you can think of potassium going in, but the key is we're going to have the cell is partially depolarized. In other words, it's closer to threshold. So it's hyperexcitable. So the sub-threshold, which wasn't supposed to fire in normal kalemia, now because you only need that much, you're going to have an action potential. But note, even though we start here, we'll kind of go back to this. Even though instead of minus 70 now, we have a resting potential here, does that mean we're going to go higher? Or does that mean now we're going to go and have the same size and shape of action potential?
Same size and shape.
Same size and shape. Very important. So although you're starting up higher so that it doesn't take as much to trigger it, because we have everything all spaced out there, once it's triggered, that's why I said same size and shape. We're going to reach the same amplitude. It's going to look exactly the same. It's just that it's going to take less of a stimulus to fire it. You're hyperexcitable because this potassium went in, made the resting memory potential closer to threshold. Hypokalemia. Our potassium levels are too low. So instead of having that amount, we only have that amount. So potassium is going to go which direction?
Out.
And make the cell more,
Negative.
which is
Below threshold.
the threshold are hyperpolarizing it even more, which is, let me show it here. Okay, kind of showing that. So, with hypokalemia, low potassium, okay, the cell becomes hyperpolarized, is further from threshold, but we'll call it hyperpolarized. Which means it's less likely to fire. So you're not going to be reacting to impulses that you're supposed to be reacting to, kind of shown here. So if this was supposed to be a stimulus in which you would react, when you're suffering from hypokalemia, you'll be less responsive. What could cause this condition? Well, of course, some kidney problem we'll talk about, because everything's a kidney.
Too much bananas.
Well, we're going the opposite way now. Now we got hypokalemia. Don't say not enough bananas, but it could be. But the idea is, let's say this is our happy cell, and we've got, potassium out here and sodium, etc. And normally our cells are happy, right? And this is what kind of solution to make them happy? It's isotonic normally, right?
Oh.
We want it isotonic normally. Now, what if you are running a marathon and which you're sweating some electrolytes, including potassium, and you start drinking pure water and pure water? You're going to do what out here? You're going to dilute out your fluid. You potentially have hypokalemia. You don't have enough potassium out here. And if you don't have enough potassium out here, and you don't have enough electrolytes out here, which way is water going to go? Where's the party?
And
The party works, man. I don't care how much trouble I get from that from my peers. Whenever you're in doubt, think of the party. Because it's really important you know the way water goes. Water is going to go in, and if the cells in your brain, that's going to hurt, okay? Because basically, you have no space. So that kind of condition, which could cause it, in which you do sort of drink too much water, it doesn't happen too much, is called hyponatremia, which is too much water. Not enough salts. You're diluting out your salts, including potassium, and people have died of that. Not many, but you might even have heard of some marathon